Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year January 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Kerala SET Biotechnology been discussed, you can go to my channel Omni Bioentrance. In that playlist, see for Kerala SET exams and there you will find. Now coming to the next question. Which of the following statement is true for trickling filter sludge? It is more difficult to thicken, that is de-water, than activated sludge. No, it is easier to thicken, that is de-water, than activated sludge. They have asked for true statements. So this is not correct. It is bulky. It is not bulky with higher sludge volume index. No, it is having low sludge volume index. It is comparatively low sludge volume index. Yes, that's correct. It has comparatively low sludge solid concentration. No, it has higher sludge solid concentration. So D is also not correct. So answer is option C. Then identify the wrong match. Detergents, lipase, textile, amylase, alcohol, nitrogenase. That is not correct. How can alcohol require nitrogenase, which is involved in nitrogen fixation? So it is not correct. Then under PFA rules, toned milk should contain dash percentage fat and dash percentage SNF. What is PFA? PFA is prevention of food adulteration. And what is toned milk? It is a blend of either the cow's milk or the buffalo milk with skimmed milk powder and water. That is toned milk. So how much fat it will contain? It will contain 3% fat and 8.5% SNF. What is SNF? Solid, not fat. That is the full form of SNF. That means milk solid contents. That is 8.5% SNF. Then dash is non-permitted food color. So it is rhodamine. Rhodamine is banned because it is carcinogenic as well as toxic. So D is the answer. Then name the bacterial spoilage causing organism in processed and cured meat products such as sausages, bacon, ham and canned meat. So Moraxella. It is associated with spoilage of poultry and fish meat, but less common in processed meat. So that is not the answer. Alkali gens, it is found in environmental samples and it is not linked to any meat products. Then Lactobacillus sake, it is spoilage organism in poultry, meat and dairy, but less specific to cured meat. No, that is correct answer. It is causing causative organism in processed cured meat products such as sausages, bacon, ham and canned meat. Whereas Pseudomonas, option D is the one which is not specific to cured meat. So C is correct. Lactobacillus sake is correct. Then next one. Which of the following is non-biodegradable polymer? So non-biodegradable means it will not be decomposed by the environmental organism, microorganisms present in the environment. So it is non-biodegradable. PLA is polylactic acid and it is derived from cornstarch, sugarcane. So naturally it is biodegradable. PHB is polyhydroxybutyrate, again produced by bacterial fermentation. So it is biodegradable. PHBV, it is polyhydroxybutyrate valerate. That is also biodegradable. But LDPE, that is low density polyethylene, it is not biodegradable, and low density polyethylene you can see in plastic bags, bottles, containers, etc. So D is the answer. Then the enzyme used in tenderization of meat. Tenderization is breaking the connective tissues in the meat to make it more palatable, softer and easier to cook. So the enzyme that is used in tenderization of meat is papain. Answer is option A. Pullulinase, it is used in brewing and baking industry. It is starch degrading enzyme. Pectinase, it is used in juices to break down pectin in fruits and vegetables. Catalase is in, it decomposes hydrogen peroxide to hydrogen and water and is used in textile industry also. Then next one, the art and science of etching, writing or printing at the microscopic level in order of nanometer is, it is called as nanolithography. Answer is option B. Then next one, in which of the following fermenters the impellers are replaced by constant flow of gas? So in airlift fermenter there is constant flow of gas to mix and circulate the culture medium, and they don't use impellers. Whereas in a tower fermenter you use impellers. Okay, in hollow fiber again impellers or pumps are used for circulation. Then perfusion bioreactor also impellers or pumps are used for mixing. So A is the answer. Then next one, the algorithm used in global alignment of nucleotide sequences, it is Needleman-Wunsch algorithm. It is for a pairwise alignment method and it finds the optimal alignment between two sequences. Then BLAST is for local alignment. Multi-align is a multiple sequence alignment tool. Then Clustal Omega is also in multiple sequence alignment algorithm. So C is the answer. The carbohydrates which are mainly present in whey, So whey, it is a byproduct of the cheese production or casein manufacture, and it is rich in carbohydrates, water, proteins, minerals, vitamins, etc. So the carbohydrate which is mainly present in whey is B, that is lactose, and it is around 70 to 80% of the total carbohydrate concentration. So the answer here is option B. The next one, the raw materials which are important for the industrial production of glutamic acid. So for industrial production of glutamic acid you require carbohydrates, that is glucose, molasses, sucrose, like that. Then peptone is required, then inorganic salts are required, and biotin is also required. And biotin concentration in the fermentation medium, it can influence the yield of glutamic acid. Why? Because Corynebacterium glutamicum, which is used in fermentation, it requires biotin for its growth. Okay, so low biotin concentration means it can suppress the cell growth, but high biotin concentration means it can block the secretion of glutamic acid by the bacterium. So you require a optimal concentration of biotin in industrial production of glutamic acid, and it is highly important. So the answer here is option D. Then next one, Primo Max is a nanotechnology product used for imparting stress tolerance to turf grass. Okay. Name the company which developed Primo Max. So it is Syngenta. Answer is option C. The next one, which of the following organism is present in natural living style as biofilms? So biofilms, they form when microorganisms attach to surface and they produce extracellular polymeric substances. These extracellular polymeric substances provide protection and facilitate communication among the various microorganisms involved in biofilm formation. So which of the following organisms represent the natural living style as biofilm? Natural living style. It is bacteria. Answer is option A. Then which one of the following is sedimentary type of biogeochemical cycle? It is a +2 question. So naturally nitrogen, oxygen, you know it is gaseous cycle. So phosphorus and sulfur are the sedimentary cycle. Answer is D. Then next one. Fermentation of glycerol in wine results in amertume. It is caused by bacteria such as Lactobacillus brevis and Lactobacillus buchneri. They oxidize glycerol to acrolein, which has a bitter taste. It is called as amertume. Then mossy flavor, it is associated with the strains such as Enococcus oni and Leuconostoc misandroids. Then they produce 3-acetyl L-pyrroline that has been associated with mousy flavor in several wines. Then tourne, T-O-U-R-N-E, it is spoilage induced by Lactobacillus brevis. Then posse, P-O-U-S-S-E, it is a gasiness resulting from liberation of carbon dioxide by heterofermentative lactic acid bacteria. So the answer here is, they have asked fermentation of glycerol, it is amertume. Then the TDT, thermal death time curve. Thermal death time curve, it is a graphical representation of the time required to kill a specific percentage of microorganism at different temperatures. That is thermal death time curve. So it is a graphical representation of the time required to kill a specific percentage of microorganism at different temperatures, and this is applicable for canning of foods. Answer is option C. Then next one. The technique used to visualize the rapid lateral movement of membrane lipids in a lipid bilayer, it is FRAP, F-R-A-P, that is fluorescence recovery after photobleaching. Now other options, FACS, that is fluorescence activated cell sorting. In the name itself it is there, used for sorting cell based on fluorescent markers. MACS, it is magnetic activated cell sorting, again used for cell sorting using magnetic beads. Then FRET, F-R-E-T, fluorescence resonance energy transfer for protein-protein interaction studies. So what is remaining? FRAP, F-R-A-P. It is used to visualize lateral movement of membrane lipids in a lipid bilayer. Then next one. The cell organelle in which detoxification of xenobiotics take place. So detoxification, it takes place in the smooth endoplasmic reticulum. Answer is option B. Then neurofibrillary tangles. What is that? They are intracellular aggregates of hyperphosphorylated tau protein. They are neurofibrillary tangles. Found in the brain cells of patients with neurodegenerative disease consist of. That is the question. So these neurofibrillary tangles, they are intracellular aggregates of hyperphosphorylated tau proteins. They are found in Alzheimer's disease, and tau is a microtubule associated protein. So neurofibrillary tangles of the brain cell of patients with neurodegenerative disease consist of microtubule associated proteins, that is tau. In your answer key it is just microtubules, that is C, but as per me it is B. Then next one. Sugar linked to the hydroxyl group of serine and threonine in O-linked glycosylation. So the first step is the addition of N-acetyl galactosamine to the hydroxyl group of serine threonine residues in O-linked glycosylation. Then this is followed by addition of other sugars such as glucose, galactose, N-acetyl glucosamine, etc. So from the options, you know, answer is option A, that is N-acetyl galactosamine. Mannose, it is involved in N-linked glycosylation, and sialic acid, it terminates the O-glycan chains. Then next one. A scientist induced a mutation on an endoplasmic reticulum resident protein folding enzyme that has lost its KDEL sequence, that is endoplasmic retention sequence. Which of the following combinations would most probable consequence of such a mutation? That means protein folding enzyme has lost its KDEL sequence. Now KDEL sequence, it is required for retention in the endoplasmic reticulum. So the protein will not be present in the endoplasmic reticulum, it will be exported to the extracellular space because it doesn't have KDEL sequence. So first one, PEF is exported to the extracellular space. Yes, that's what I told you just now. PFE is degraded in the endoplasmic reticulum. It is not degraded. Its retention is affected. So 2 is not correct. Unfolded protein increase in the endoplasmic reticulum. Yes, that's correct because PFE is a protein folding enzyme. So unfolded proteins will accumulate in the endoplasmic reticulum in the absence of PFE. So 3 is correct. Then PFE is transported to the cytosol. It is not to the cytosol, it is to the extracellular space. So 1 and 3 is the answer. Answer is option C. Then hybrid dysgenesis is caused by which of the following transposable element? It is P element, it is caused. And what is this hybrid dysgenesis? It occurs when male with P element is fertilizing a female that lacks the P element. The resulting offspring will be sterile male and female. That is called hybrid dysgenesis. So the answer here is P element. Then next one. A woman with gene for hemophilia and a gene for color blindness on one of her X chromosomes. So one X chromosome is having gene for hemophilia and gene for color blindness. Marries a normal man. So her second X chromosome doesn't have hemophilia and color blindness genes in it. So she is a carrier of both hemophilia and color blindness genes. How will their progenies become? So this is the female, HC is hemophilia and color blindness. On one X chromosome, the other X chromosome doesn't carry the genes. Marries a normal man. These are the gametes produced. These are the offsprings. So if you see, out of 50% sons, here is 150%, here is the other 50% son. 50% of the sons will be hemophilic and colorblind and 50% will be normal sons. Whereas daughters, they will not have the disease, but they will be 50% will be carriers and 50% will be normal. Hemophilic and colorblind daughters, no. All sons and daughters are hemophilic and colorblind. No, daughters are carriers. Sons, only 50% are affected. 50% hemophilic and colorblind sons and 50% normal sons. That's correct. 50% hemophilic sons, 50% colorblind daughters. That's also not correct. So the answer is C. Then next one. Which of the following ensures stable binding of RNA polymerase at the promoter site? Rho factor. Rho is involved in transcription termination. So not binding of RNA polymerase. MCM proteins, they are essential for DNA replication. Sigma factor, yes, that ensures stable binding of RNA polymerase at the promoter site. Rec proteins, they are involved in DNA repair. Then how many calcium ions could be bound to calmodulin protein? So calmodulin, it is involved in calcium signaling, and calmodulin, it has got two globular domains. How many? Two globular domains, and each domain contains two EF hand motifs. So each domain, it contains two EF hand motifs. So two globular domains. One globular domain is having two EF hand. The second globular domain is also having two EF hands. And each EF hand motif, it binds to one calcium ions. So totally 4 calcium ions will be bound by the calmodulin protein. So the answer here is option D. Then next one. Arrange the following sequence of extracellular signaling in the correct order. So signaling cell synthesize and release signaling molecules. That will be the first one. Then comes transport of the signal to a target cell. Yes. Then binding of the signal to a specific receptor. Yes. Start of the signal transduction pathway. Yes. So the option will be 3, 1, 4, 2. 3, 1, 4, 2 is your option B. Then human mitochondrial genome encodes for dash rRNA genes. So mitochondrial genome, it encodes for 2 rRNA genes, that is 12S rRNA and 16S rRNA. So answer here is option C. Then next one. Which of the following would cause Hardy-Weinberg principle to be inaccurate? Okay, large population size, that is a requirement of Hardy-Weinberg. Individuals of the population mate in random, again Hardy-Weinberg equilibrium requirement. Natural selection is present. Yes, that is not required in Hardy-Weinberg equilibrium. So they have asked which will cause it to be inaccurate, that is natural selection is present. There is no source of new alleles from outside the population. That's also Hardy-Weinberg requirement. Then next one. Which of the following about retinoblastoma, that is RB, tumor suppressor protein is correct? Now retinoblastoma, it regulates the cell G1S transition of the cell cycle. That means it prevents premature cell division and maintains genomic stability. Now what is the mechanism behind this RB tumor suppressor protein pathway? So first is unphosphorylated retinoblastoma. They bind to E2F transcription factor and prevents the transcription and thereby prevent the cell from entering the S phase. Okay. Now when a mitogenic signal is received, it will activate the CDKs. The CDKs, they will phosphorylate the retinoblastoma, inactivating it. Now once the retinoblastoma is inactivated, it will be release the E2F transcription factor. The free E2F transcription factor, it will promote transcription and cell cycle will progress. So this is the pathway. Now first option, it is activated when phosphorylated by CDK. It is inactivated when phosphorylated by CDK. So A is not correct. It binds E2F transcription factor and prevents cell from entering S phase until a mitogenic signal is received. That's correct. What is a mitogenic signal? Mitogenic signal is that signal that stimulates cell proliferation, differentiation, growth, etc. That is mitogenic signal. Then it is a transcription factor. So retinoblastoma, yes, it is not a transcription factor. It binds to E2F transcription factor. So C is also not correct. When a mitogenic signal is received, it binds the transcription factor E2F and thus stimulates the cell to enter S phase. It doesn't bind when a mitogenic signal is received to E2F transcription factor, but it releases the E2F transcription factor and thereby stimulate the cell to enter S phase. So D is also not correct. Then next one. The function of UVRC in nucleotide excision repair mechanism, that is NER, is. So this is the damaged DNA site. UVRA and UVRB protein, they will bind to this damaged site. UVRA is a dimer, UVRB is a monomer. Then once they are bound, the UVRA dimer will be dissociated. Only UVRB will be bound to the lesion. This will cause the binding of UVRC at the damaged site. UVRC binds to UVRB. Then what will happen? UVRB make an incision at the 5th phosphodiester bond on the 3' side of the lesion. That is on this side. This side somewhere it will make a lesion. UVRB. Now UVRC will make a lesion on the 5' side at the 8th phosphodiester bond. The resulting 12 to 13 nucleotide fragment will be released by what? UVRD helicase. That is this UVRD helicase. It will remove the 12 to 13 nucleotide fragment, and this gap, it is filled by DNA polymerase 1, and DNA ligase will seal the ends. That is what is happening in nucleotide excision repair mechanism. So the function of UVRC they have asked. To identify and locate lesions in DNA. No. That is done by UVRB and UVRA. After UVRA dissociates, leaving UVRB DNA complex, then only UVRC is binding. So A is incorrect. Then to act as a helicase to unwind the site, it doesn't act as a helicase. To catalyze the incision at the 5' end of the lesion. Yes, that's correct. To ligate the strands, ligation is done by DNA ligase. So C is the answer. Then next one. In Drosophila males, there is complete linkage. What is the reason behind this? Direct question. It is lack of synapses. Because Drosophila males, they have only one X chromosome and the other is Y chromosome. So recombination not possible. Then chiasmata are first seen in. They are first seen. They have asked first seen. They are first seen in the diplotin stage. Answer is option B. At the end of each phase of cell cycle, cyclins activating CDKs in that phase are inactivated irreversibly by. So they have asked how are cyclins deactivated? That is the question. So in cell cycle regulation, these cyclins, they bind to CDKs. They will activate the CDKs, drive the cell cycle progression. Now cyclin degradation, it will terminate the CDK activity. So how is this cyclin degraded? That is by tagging with ubiquitin to cyclin. Cyclins are proteins. So ubiquitin tagging. Then proteasomal degradation. That is after ubiquitinated cyclins are degraded by the proteasomes. Multiple phosphorylations. That regulates the CDK activity, not degrade the cyclins. So A is incorrect. Dephosphorylation. Again it doesn't degrade the cyclins. Ubiquitination. It only tags the cyclin for degradation, not degrade them. Destabilizing by proteolysis in a proteasome, that is degradation. So answer is option D. Then next one. Prenylation adds prenyl group to the dash amino acid residues. It is cysteine. Answer is option B. Which of the following inactivates the 60S subunit of eukaryotic ribosomes? So it is ricin. Answer is D. Chloramphenicol, it binds to 50S subunit of prokaryotic ribosome. Diphtheria toxin, it is ADP ribosylates elongation factor EF2. So ricin is the answer. Then the type of topoisomerase that can introduce negative supercoils, it is type 2 or DNA gyrase. Answer is option B. Then which of the following are reducing sugars? Sucrose. Sucrose is a non-reducing sugar because, okay, what is a reducing sugar? Reducing sugars, they can donate electrons through their aldehyde or ketone group. They have free aldehyde ketone group. Whereas sucrose, the bond is such that there is no free aldehyde and ketone group. That is because the anomeric carbon of glucose 1 is bound to the anomeric carbon of fructose, that is C2. So this involves a glycosidic bond between the glucose and the fructose. Thereby there is no free aldehyde and ketone group present. So sucrose is not a reducing sugar. Then ribose has got a free aldehyde group. Fructose is having a free ketone group. So they are reducing sugars. 2 and 4 are reducing sugars. Lactose, again a disaccharide, but it has got a free aldehyde group. So this can be oxidized, and which allows lactose to act as a reducing sugar. So 2, 3, 4 only. Answer is option B. Then a molecule of amylopectin which contains 1200 glucose residues and is branched after every 40 residues. How many reducing ends are there? So no matter how many residues are there, but a molecule of amylopectin has only one reducing end. So answer is option B, 1. For a protein with 100 amino acids, how many possible sequences are there? So there are 20 amino acids. And each position in that particular sequence, it can be occupied by any of the 20 amino acids. So the possible number of sequences is 20 raised to 100. Answer is option D. Then which of the following is involved in pentose phosphate pathway? Glucose 6-phosphate dehydrogenase. Yes, that is involved in pentose phosphate pathway. And glucose 6 dehydrogenase, it catalyzes the reaction of glucose 6-phosphate to phosphogluconolactone. This is the first enzyme. Glucose 6-phosphate dehydrogenase is the first enzyme in pentose phosphate pathway. Acyl-CoA glycerol transferases, they are involved in lipid metabolism. PFK2-FBPase, that is phosphofructokinase 2, fructose 2,6-biphosphate, it regulates glycolysis and gluconeogenesis. Pyruvate dehydrogenase, it converts pyruvate to acetyl-CoA and a link between glycolysis and citric acid cycle. So if you know the action of B, C, D enzymes, then you can happily mark the answer as A. Then the lipid which act as lung surfactant. Now what are these lung surfactants? They are a mixture of lipids and proteins lining the alveoli of the lungs, and it allows the lungs to expand and contract easily during exhalation. Okay. So they have asked the lipid which act as a lung surfactant is phosphatidylcholine. Answer is option C. Ceramide, it is involved in apoptosis, cell signaling, etc. Phosphatidylethanol, it is found in cellular membranes involved in cell signaling. Phosphatidylinositol also in cell signaling. So that's it for now. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of Kerala SET Biotechnology been answered, you can go to my channel Omni Bio Entrance. In the playlist, select for Kerala SET Enzymes and there you will find. Thank you.